Thank you very much, the organizers, for inviting me to talk. We got on to doing cancer research, and we're really looking at the role of glycosylation in some of the growth factor receptors. And we identified that the glycosylation of these growth factor receptors play a critical role in terms of the multi-stages of tumorigenesis. So one of the highlights that in my talk here is to look at this innovative, promising, entirely new targeted therapy for cancer. And what we identified is a whole new signaling paradigm that seems to regulate the receptor tyrosine kinases, in particular, for example, the EGFR, which plays a very prominent role in cancer development. These particular RTKs are highly glycosylated receptors. And in particular, we also looked at toll-like receptors. This is more in tune to the immune-mediated tumorigenesis. And this is as a result of pro-inflammatory cytokine production and that may develop into tumor neovascularization and tumor development. And so what we did is we identified that these receptors, the receptor tyrosine kinases and toll-like receptors, are highly regulated by this whole new signaling paradigm that we discovered. And this is in relationship to this mammalian neuraminidase 1 called NEU1, which is in complex with matrix metalloprotease 9. And matrix metalloprotease 9 plays a critical role in a lot of the tumorigenesis, or multi-stages of tumorigenesis in terms of its proteolytic activity. And also in addition to a G protein coupled receptors. So there is this three-part type of receptors that play a critical role as in this multi-stage of tumorigenesis. The predominant feature is that neuraminidase 1 cleaves the alpha-2,3-sialic acid, which facilitates receptor association, dimerization, and signaling. And this is how this whole manifestations of tumor development is manifested. Our preclinical animal studies support an entirely new cancer targeted therapy. In relationship that this particular targeted therapy is unaffected by a lot of the mutations in the growth factor receptors. This is very true for the EGFR receptor and some of the other kinds of growth factor receptors like fibroblast growth factor, insulin, and so forth. This targeted therapy also blocks tumor neovascularization. It overcomes chemoresistance of tumors. This is a very important area in cancer. I was at a meeting at the American Association for Cancer Research, and one of the cries out for study is why cancers develop chemoresistance, and trying to really understand why this is happening. The end result of the chemoresistance is that they develop metastatic disease as a result of the treatment. This particular cancer therapy blocks immune-mediated tumorigenesis as a result of some of the pro-inflammatory cytokines that develop neovascularization and metastasis in tumor growth. And as a result, it also can block the invasion and metastasis of cancer. So just to give you a pictorial illustration of what we're looking at here, this is a representation of the receptor tyrosine kinase. We've looked at EGFR receptor, the TRAC receptor, which is the nerve growth factor TRAC-A. We also find it for the brain-derived nerve growth factor. We've also looked at the insulin receptor. These are the receptors that are highly glycosylated, and they're represented as receptor tyrosine kinases. What we found, which was quite interesting here, is that this neuraminidase 1 forms a trimeric complex with the protective protein cathepsin A and the elastin binding protein. It's the elastin binding protein that keeps the neuraminidase 1 from being active. This has to be in complex with the cathepsin A in order to become active. So one of the things that we found here is that when ligand binds to its particular receptor here, what happens is that there is evidence to show that these go through a conformational change. That conformational change activates the GPCR. And we found that the GPCR is this neuraminidin B, which is, for example, the bound-basin receptor that forms a complex with the tyrosine kinase receptor at the actal domain. When there's this, do you have another pointer? Oh, just wait, I've got this, I'm sorry. Okay, so when there's a conformational change here, the GPCR goes through activation. This activation activates the matrix metalloprotease 9. Matrix metalloproteinase, because it's complex to the active domain of the receptor, removes the elastin binding protein because it has that substrate specificity. As a result of that, NEU1 becomes activated. And what we found here is that NEU1 has specificity for the alpha-2,3 sialic acid. This is the terminal sialic acid on these highly glycosylated receptors. And as a result, once sialic acid is removed, this facilitates dimerization, signaling, and you get this whole manifestation of tumor development and the whole aspect. We found that this particular signaling paradigm plays a critical role in tumor growth, metastasis, tumor neovascularization, and chemoresistance. And what we've looked at here is the highly metastatic and highly aggressive pancreatic cancer. We looked at the triple-negative breast cancer as well as ovarian. And here we looked at the heterotopic xenografts of tumors growing in this RAG, common gamma, double mutant mice. These particular mice have a deficiency in T cell, B cell, but in particular has a deficiency in NK cells. The NK cells do play a critical role in preventing metastasis if they're present. So what I'm going to do for you here is that I'll present to you some of the highlights of some of the papers that we've published on this particular area. And in one particular paper here is looking at the epidermal growth factor receptor in terms of this novel signaling paradigm and how it's translated to pancreatic cancer. And I'll look at sort of snippets from this particular paper to give you sort of an idea of the direction we're going here. As you know, the EGFR here is a fully glycosylated receptor, and it has 8 of the 11 conical end glycosylation sites. And as you can see here from published papers, 2 of the sites are non-glycosylated and 1 is partially glycosylated. So one of the fundamental questions here, what is the role of this glycosylation in terms of receptor activation? And so a lot of, up to this time, the mechanism behind epidermal growth factor-induced receptor activation was unknown. All we knew that the receptors have to dimerize in order to become, provide the signal to the cell to do a particular function. So basically, we asked the question whether modification of the glycosylation of the EGFR receptors involves this activation of neuraminidase 1, which we found to be complex at the active domain of the receptor. And so what we did is looked at this in a very careful way. We did cell studies in terms of colocalization using confocal microscopy. We did immunoprecipitation. We also did it at the genetic level and gene knockout or siRNA knockouts for some of these key players. And we found that the neuraminidin B receptor, which is called the NMBR or BB1, which is called now, it forms a complex with neuraminidase 1 and MMP9. And these 3 compounds, important compounds, play a very critical role at the ectodomain of the receptor, EGFR receptor. And this has been all done by all these different types of techniques. Just to give you sort of a feel for how this may be manifested here, here we looked at PANC1 cells, MIA-PACA2. These are pancreatic cancer cells in culture. And one of the things that we asked the question is that if neuraminidase 1 is activated as a result of ligand binding to the receptor, when we add to these live cells a substrate for neuraminidase 1 that surrounds the live cells, then if this is active, then this particular substrate called forminato would be cleaved to its methylimbellaferone, and this will give you a blue color under fluorescent microscopy. And sure enough, when we add EGF to these particular PANC1s, we get this sialidase activity, which is manifested as a result of Neu1 being active, cleaving the substrate to give you this blue color. When we add, for example, we looked at a variety of different neuraminidase inhibitors, and we found, and we published on another paper that this osmivir phosphate, or generic name called Tamiflu, when we add the Tamiflu to these live cells in presence of EGF, we're able to block this activity. So it seems that this osmivir phosphate Tamiflu inhibits this activity when it's in the presence of EGF. We've looked at anti-Neu1 antibody. We pre-treat these cells with anti-Neu antibodies, and we can block this Neu1 activity. And we also added the inhibitor specific for the MNB matrix metalloprotease 9, and we're able to block this whole activity. So when ligand binds, even goes through the GPCR, if this is blocked, then you don't get this activity. We've looked at PANC1, MIA-PACA2, and we get the very similar kinds of reaction patterns. All of this also been looked at. For example, we were able to do a gene knockdown or siRNA knockdown in terms of matrix metalloprotease 9, or even the GPCR for neuraminidase B, and we're able to show that we are unable to get this kind of activity. So moving forward in the animal studies is that we use these MIA-PACA2 pancreatic cancer cells, and we did a lentivirus transfection with the EGFP. So these cells under biophotomic imaging glow beautiful green, so we're able to locate whether we have tumor within the animals. So when we did the tumor growth over a period of time up to 50 days post-implantation, we found in the black here, untreated animals, there was a significant increase in growth of tumor, shown here. And we started the treatment with osmivir phosphate at this point here around day 42 post-implant, and we're able to block any tumor growth. And you can see a very small tumor growth here. We did tumor size, and we looked at tumor weight, and we showed that the treatment with osmivir phosphate, 100 mg per kg on a daily basis, significantly reduced the tumor size and tumor weight. One of the interesting things to show here is that this particular cancer, these are live necropsy tumors. We were able to excise the skin, and we were able to see a very extensive tumor neovascularization, blood vessel formation. But when these animals were treated with Tamiflu, we saw a very small amount of this blood vessel formation, which was quite exciting in terms of here's the possibility that we may be able to block this tumor neovascularization. If that's the case, then we may be able to block metastasis. Just to really solidify what we're looking at here is that, we took individual necropsy tumors were taken from the untreated as well as the Tamiflu-treated animals, at the end of the experiment, we were able to lyse the tumors, individual tumors, and containing the protease and phosphatase inhibitors. And here we looked at the phosphorylated EGFR using Western blot, or phosphorylated STAT1, or even phosphorylated or activated NF-kappa B. And so when we looked at this, as you can see in the bar graph here, these are densitometry of the bands here in the Western blot, and this is represented as a ratio of phosphorylated EGFR to beta-actin. And you can see here that Tamiflu treatment at the end of the experiment for all of these animals here showed a significant decrease in EGFR, phosphorylated EGFR, phosphorylated STAT1, and phosphorylated NF-kappa B. So this gives us an indication that this particular treatment targets these xenografts of the myopathic tumors growing in these double mutant mice. Since we had the lentiviral transfection of the enhanced GFP in these myopathic cells, we are at the time of necropsy, we looked at these live necropsy tissues using biophotomic imaging. And as you can see here, A here is the tumor. And because it's been looking at the black and white pixels, the tumor, the EGFP, the myopathic cells would be shown here as a black. It's here, the tumor here is black, but we also see here that the B here is the liver. So we have quite an extensive type of metastasis to the liver. And so when we did the treatment here, we see very little metastasis to the liver. We also looked at these necropsies here. We did the H&E staining, and we were also looking at, for example, metastatic clusters within the tissue of liver, and we were able to count all of these metastatic clusters. As you can see here, the untreated control had quite a bit of metastatic clusters, whereas the Tamiflu treatment here was significantly reduced. We also looked at, for example, the lung, and here, as you can see here, here's a metastatic cluster in the lung. You don't see the nodules in the lung, but within the lung, you see this cancer. And we started counting these, and as you can see here, the treatment here had a significant reduction in the metastatic clusters in the lung. I just showed you a snippet of that particular study showing that the efficacy of this kind of a treatment does have a powerful play in terms of preventing tumor growth, tumor neovascularization, as well as metastasis. The other important question here is, does this play an important role in chemoresistance? And so we published this paper here that we were using this therapeutic targeting of this neuraminidase 1, as you can picture that particular signaling paradigm with this osmolar phosphate. And we published this paper showing that this particular compound is able to disable cancer survival in this pancreatic cancer with acquired chemoresistance. The clinical problem here is that the resistance to drug therapy relates to very high rates of metastasis in patients, and this contributes to the low survival rate in patients diagnosed with pancreatic cancer. The major problem here is that chemotherapy causes the cancer to become chemoresistant. In that process of chemoresistance, it changes the phenotype to mesenchymal, and it starts spreading, and it develops into metastatic disease. Just to give you an idea, one of the things we did here in order to establish chemoresistance of this PANC1 pancreatic cancer cells in culture, we were able to establish these chemoresistant cells against this gemcitabine at 0.1 micromolar, and these grew beautifully in culture over a year. So we established this long-term chemoresistance against this gemcitabine. Gemcitabine is the standard chemotherapeutic used on patients in the clinic. And so here's an example where we used gemcitabine at different dosage, and it had no killing effect. But in these chemo-resistant pancreatic cancer cells, in the presence of increasing dose of osmometer phosphate, we can get a significant decline in cell viability. And when you look at these doses here, we're able to get an IC90, a very significant decrease. The IC90 is, for a clinical outcome, is very, very significant in terms of survival of the patients. We looked at another chemotherapeutic called cisplatin. These cells grew in cisplatin about 80 micromolar over a year. They just love to grow in this particular chemotherapeutic. And when we did the treatment with osmometer phosphate, we're able to get a very significant decrease in cell viability. We also looked at combination of cisplatin and gemcitabine. These cells grew beautifully in this two chemotherapeutics over a year. And we did the treatment here, we got a very beautiful IC90 at a much higher dose. One of the things we found in the animal studies is that osmometer phosphate, or Tamiflu, prevents, there's no toxicity. The animals were able to tolerate this very well. Osmometer phosphate had very limited cell viability or targeting normal cells. We looked at peripheral blood mononuclear cells from patients, and we found that it has no high viability. It was not toxic. And we also did some chemotherapeutic combinations with Tamiflu with standard chemotherapeutics in the animals. We found that it prevented any hypersensitivity reactions or pro-inflammatory. A lot of the chemotherapeutics used in patients, the patient become hypersensitive, and this is where the patient gets taken off the therapeutics. In combination, we're able to prevent all that happening. One of the other things that is quite important here is that the chemotherapeutic causes the cancer to become, changes this EMT, which is epithelial mesenchymal transition. The EMT transition in cancer, especially the tumor, when it becomes EMT, it develops into metastatic disease. And so when we did the study here on PANC1 untreated, we looked at E-cadherin, which is a marker for the epithelial. The N-cadherin here is more related to the mesenchymal type phenotype, which is metastatic. And here, when we did the treatment here of these PANC1s, we can upregulate the E-cadherin and downregulate the expression of N-cadherin. So when you look at when we measure all of these different staining properties here, we can see here there's a significant decrease in the expression of N-cadherin and V-E-cadherin, which is a property or a biomarker for mesenchymal phenotype of the cancer, which is more metastatic. This is very similar. We looked at the PANC1s with gemcitabine resistance, so grown over a year, and we can show the same effect. And this is also true with cisplatin resistance, a very similar response. And so one of the other questions here is that, is this related to the tumor itself in these animals? And so we were able to, for example, look at the necropsy tumors, and we stained for E-cadherin, N-cadherin, V-E-cadherin. Here we used, in this particular group, a cohort, we treated these animals with a very low dose of Tamiflu. This should be, for example, this is 2 milligrams per kg. This is just representative of one tumor, necropsy tumor. And when we looked at the treatment here, we see a very strong staining for E-cadherin. Gemcitabine here treated animals at 30 mg per kg. We see very little E-cadherin, but we still see quite a bit of the N-cadherin, which is more mesenchymal metastatic form of the cancer. And when we did the combination with gemcitabine with Tamiflu at these different dosage here for this particular, we're able to revert this to E-coherent and less metastasis. So this gives us an indication that here's a treatment that can reverse EMT and prevent metastatic disease. Oops, sorry. So we looked into the triple-negative breast cancer, and as you can see here, these breast cancers lack the estrogen, progesterone, and this epidermal growth factor receptor 2, HER2 neuron receptors. Very difficult cancer to treat in patients with this particular triple-negative. These particular patients have typical high grading. They're more frequent relapses, and they exhibit poor outcomes, and the prognosis is very, very limited. Patients die from this. Currently, there are no targeted therapies that are effective for triple-negative breast cancer. I'm just giving you just a snippet of some of the results from this paper, which is quite interesting because this particular kinds of treatment, as you can see here, which is quite interesting, is that when we did the live necropsy of the tumor, here you can see the untreated animals, there's extensive blood vessels. Very, very extensive. This particular, this gives me indication that this particular tumor or cancer cells has this very high propensity to develop neovascularization, blood vessels, so they can metastasize quite readily. Here we did a cohort of animals were treated with osmomer phosphate at 30 mg per kg. And here you see sort of the tumor volume. And here's one animals that seem to not respond well, whereas some of these other ones did respond to the treatment compared to the untreated. Here you can see here that the OP at 30 mg per kg did reduce the tumor neovascularization considerably compared to the untreated. Here, which was quite interesting, that osmomer phosphate at 50 mg per kg, here showing that this is the tumor growth, and here we went up as high as 150 days. Here we had to stop because some of these animals did develop quite a bit of lesions on the skin, and we, according to animal care, we had to sacrifice these. These animals that treated with 50 mg per kg OP, we had long-term survival, and at about 120 days post-implant, these animals were taken off the drug, and we found that there is no relapse, the tumor went away. And here you can see at necropsy, no tumor recall. Okay. Here we looked at metastasis. Untreated, there is extensive metastasis here for the untreated control in these lungs. We didn't see much of the nodules, but within the lung here we see metastasis. When you did the treatment with different dose of OP, we got a significant decline, as you can see here. This to highlight the ovarian cancer was a very important area here, and we were interested to see whether the snail, which is a transcriptional factor, plays a critical role in this epithelium-mesenchymal transition. It's a transcriptional factor repressor of E-coherent, so there is a relationship here, and it also is involved in tumor progression and metastasis. So we looked at this in terms of lentiviral transfection and knocking down the snail, and to see whether this in the human A2780 ovarian epithelial carcinoma cell line in these animals. And what we saw here is that this particular ovarian cancer developed very massive bloody tumors, extensive neovascularization. We looked at the slug KD, and we were unable to reduce this particular neovascularization, but when we knocked down the snail, we were able to show no tumor growth. except for one here that very small, but all the animals had no tumor growth at all. And we terminated this animal here because this kind of tumor developed lesions on the skin, and according to animal care, we had to sacrifice. So basically, the end result here is that snail here plays a critical role, and this has been published data that upregulates MMP9. So there is that relationship that snail does play a very important role in upregulating MMP9. And as you can see in our signaling paradigm, MMP9 plays one of the key players involved in activating neuraminidase 1. Knock down the snail here, you don't get the expression here, and you don't get this activation of NEU1. So the end result here is that this signaling paradigm produces signaling cascade that could be involved in tumor vascularization, secreting cytokines. It prevents tumor neovascularization, tumor growth, tumor metastasis, and we also show that it plays a very critical role through this signaling in terms of establishing chemoresistance. Shutting down NEU1 here, which plays a very important role in cleaving alpha-2,3-sialic acid, facilitates dimerization. Shutting this down, you prevent any of these manifestations happening. So we were quite pleased to see this. And so, as you can see here, this is a parsimonious model here. One enzyme that seems to regulate the majority of the growth factor receptors that we looked at. We also looked at the toll-like receptors, chemoresistance, metastasis, and angiogenesis. It plays the multi-stage of tumorigenesis, targeting one target with one drug. We can target the whole multi-stage of tumorigenesis. And I thank you very much for your patience.
Yes. Yeah, we're going to take one pressing question because we are about a half hour late. I didn't realize how late we were. I'm not being a very good moderator here, so go ahead.
Actually, I have many questions, but this is exciting.
We can have it over.
We got a lot of
exciting.
beer time.
Beer time.
I think this
beer
is.
time.
Are you going to suggest that everybody start to pop in Tamiflu?
No.
Good. Yeah. We'll talk about Tamiflu later. Yeah.
No. One of the things that has to happen, and I've been trying to go to clinical trials. One of the things, if you pop Tamiflu, it goes through the GI tract, and it the active component, which is osmium phosphate, gets catabolized by the liver enzymes to the carboxylated form. And we found that the carboxylated form of osmium has no effect on NEU1. So this particular treatment would have to be a soluble form of osmium phosphate, probably perfusion through the bloodstream.
Yeah, it's a different formulation. Okay.
It's a different formulation.
All right, we'll move on, but thank you very much, Myron.
Okay.
We'll try to talk later on.
Okay.
Thank you again.